We're underway whenever you are ready.
All right. Thank you guys for joining our presentation today. I hope this is informative. This is certainly not as thorough as it could have been. So obviously, if you have any questions as you go through, please let us know and we'll be happy to answer. My name is Brian McCullough. I'm an assistant professor here at Husson with the College of Pharmacy, or the School of Pharmacy. And I'll let Jay Singletary introduce himself when he gets to his part. We're tag teaming this today, so I'll be doing the first part of COVID-19 vaccinations. So I totally drafted this whole poster from scratch, came up with the idea and everything. But no, when I was thinking about COVID vaccination, the thing that I thought of was hope, because things are pretty bad right now. If you look at the metrics, COVID-19 is really going crazy across the country.
Sad.
Yeah, it is sad. Maine's bad, but Maine's not nearly as bad as other places. California, I'm sure you've seen in the news. But vaccination will hopefully help us get back to normalcy. So some disclosures, I have no financial conflicts of interest. However, I do have a teething baby, so sleep deprivation may retract me from the quality of this presentation. And also I like memes and pictures, so you'll see those coming up in this presentation. Some terminology that you may have heard of with COVID-19. And you may have been confused potentially if you hear one versus the other. I'm not going to be talking about all of these terms today, but I might, for instance, interchange SARS-CoV-2 and COVID-19. The difference between these two is that SARS-CoV-2 is the novel coronavirus. That is the name of the virus itself. The disease it causes and the pandemic that has been associated with it is called COVID-19. Which is coronavirus infectious disease found in 2019. And this is the pandemic caused by the virus SARS-CoV-2. SARS-CoV-1, or just SARS, you guys may remember back in the mid-aughts, where SARS was a problem that came over from Asia as well, but was pretty rapidly contained. And actually that virus mutated pretty quickly to fall out of the news cycle pretty fast because it became a lot more mild. The SARS-CoV-2, its mutations don't seem to be making it any less lethal, and in fact, make it potentially more transmissible. We're not talking about those variants today, but you may have read about those in the news as well. Some other terms you might hear: PCR, polymerase chain reaction. This is actually, it's usually a DNA test used for identification. This is actually a typo. For coronavirus, it's actually an RNA test, a test for the RNA sequence. You may also see this referred to as nucleic acid amplification testing, or NAAT, and those are interchangeable. Cycle threshold is the number of PCR cycles to exceed a predefined threshold of fluorescence to increase the reliability of PCR test. Those who are in the science fields, you'll know that any instrumentation has background noise. And so these cycle threshold counts are set above that noise level so that when you have a signal, it's much more likely to be the target that you're interested in this case, the RNA of SARS-CoV-2, and less likely to be background noise. And the higher the cycle threshold, the more times that PCR has to run, the lower the amount of DNA or RNA. So the lower amount of genetic material that's in that system. So higher threshold, or higher cycle count means lower level of virus generally. And then mRNA we'll talk about today is messenger RNA, and I'll go into that in a little bit more depth. So SARS-CoV-2 was identified in the Wuhan province of China in 2019. It was of zoonotic origin, probably from a bat, is what they're thinking. And it's related to the SARS virus back in the 2004-2005 era. And so hence the name SARS-CoV-2. It produces a wide array of symptoms and severities. The common symptoms are fever, cough, anosmia, or the inability to smell or taste, and fatigue. And it may lead to pneumonia, adult respiratory distress syndrome, or ARDS, and potentially even death. Here is just a fancy schematic of all the different lineages of SARS-CoV-2. There have been actually quite a few variants that have been identified to this point, but none of them have been as problematic as the variants that were found in the UK and in South Africa, which are not reflected on this particular slide. So the SARS-CoV-2 stages of infection. So how SARS-CoV-2 actually gets into our cells is it binds to a receptor called ACE2. And how it binds to that receptor is by the spike protein. You guys may have heard about the spike protein in the news as the target for vaccination. And that's why I'm bringing this up. But the spike protein is how the coronavirus actually gets into the cell. And so the spike protein fits nicely into this ACE2 receptor. Once that happens and the receptor binds, then the virus starts to uncoat and it releases its genetic material inside the cell. Once its genetic material is inside the cell and it's an RNA virus, that RNA is then transcribed and the proteins to make a new virus are then done. It uses our host machinery within the cell, uses the ribosomes to make proteins. It also transcribes and translates new RNA, and then that all packages together and you get more virus that comes out of the cells. And so the spike protein is really that portal of entry. Think of it like the key in a lock and key mechanism to actually unlock the entry into the cell. Some quick facts about COVID-19 is that it does have a pretty long incubation period. One of the reasons why this is probably transmitted by so many people is that transmission can occur while patients are asymptomatic, and that asymptomatic period can last as long as 14 days, with an average of about 5 days. So if you think about getting an infection, starting to replicate virus, not even having symptoms until maybe a week into the infection, you might be spreading this infection around to people and not even know you're infected. After the disease onset, then it goes to either mild or severe disease if you're not asymptomatic. Of the people who get symptomatic disease, about 14%, 14 to 20, depending on the reference you look at, have severe disease, including needing an ICU stay. These patients also will have shortness of breath, et cetera. And then about 5% of patients overall are critically ill. And many of those who make it to this stage actually don't make their way back out of this stage. This includes severe lung disease, acute heart injury, and multi-organ failure. So the age is the biggest risk factor for severity and death. Children and young adults are likely asymptomatic. So you think about Husson, we have a large potential to have asymptomatic spread here because younger individuals are less likely to have symptoms than older individuals. However, these patients may still be, folks may still be infectious. This is transmitted by droplets. And so that masking that we do, and in the hospital, we actually wear eye protection as well. This protects us and the patient from droplets expulsion. The virus is also excreted in the feces, and whether this is a means of transmission is unknown, but it does allow an interesting monitoring mechanism. And I think there are certain spots around this area. I think UMaine might be one of them. And I want to say the city of Bangor might be doing this too, of actually monitoring sewage for SARS-CoV-2 presence in there. And if they start seeing an increase, that usually is the precursor for having the community have severe disease. So that's kind of interesting. So a brief look at the last year. This maybe seems like a decade or so. It certainly does to me. It feels like I don't even remember what life was like before COVID-19. I think I remember going to restaurants. Pretty sure I went to movie theaters at some point, but that seems like a distant memory. This was the first recorded case was in December of 2019. And then in January, it was basically WHO at the end of January declared a high alert for China specifically. And then in February, increased it to a very high alert on the global level. And as of October, there were over 1 million deaths worldwide from COVID-19. And so where are we now? If you look at the map of the United States, this was taken 2 days ago, we are all in the lower 48 in some various shade of red. So in Maine, we're a little bit of a lighter shade of red, which is the active or imminent outbreak. And then a lot of the southern and western states are in a severe outbreak scenario. Interestingly, the Hawaii and Puerto Rico are a little bit lower risk. And then I believe that's the Mariana Islands are green. So, we have places we could potentially go, except they won't take us. But if we could, they would have low COVID risk. Here in Maine, you can see on the right that our incidence has been increasing. This is largely in part because of the holidays and the resultant spread from that. We are starting to see some signs that maybe our cases might be trending down. So I hope that that's true because right now we're in a pretty bad way. All right, so let's talk about the vaccination for SARS-CoV-2. So the COVID-19 virus, SARS-CoV-2, you can see these little red triangle-y things are the spike proteins. And those are what really most of the vaccines are going to target. So mRNA vaccines, they use host machinery to amplify the production of antigen and non-structural proteins required for RNA amplification. So prior to COVID-19, vaccines using mRNA technology really hadn't been done very much. There are some cancer technologies that use mRNA, but really for vaccinations, animal testing, and some very small human trials are about all we had. So this was a bit of an unknown going into it. Fortunately, as Mr. Singletary is going to talk about, is that they have both been shown to be safe and effective, which has been great. In fact, I've had 2 doses now of the vaccine, being a frontline worker, and things are going pretty well. I will say the second dose was be prepared with some ibuprofen, but other than some about a day of flu-like symptoms, these vaccines have been quite safe and very, very effective. So the mRNA vaccine for COVID uses basically our own body to produce spike protein. And what it does, it has these little snippets of viral mRNA, which are the instructions to make the protein. For both the Moderna and Pfizer vaccine, they put these in a little lipid coat, and that lipid coat helps it to get into cells pretty easily. Once it's in the cells, the mRNA finds the machinery of the proteins in the body, which is the ribosomes, and those ribosomes start kicking out a bunch of spike proteins to a pretty great extent. Make a lot of spike protein in our body. But these spike proteins are harmless on their own because all they are just the mechanism for the virus to get in. There's no genetic material contained in this, so there is no risk at all of a virus being made from this mRNA. The only instructions that it gives the cell is to make spike protein. And the resulting immunity so far has shown to be very robust. So to talk about spike proteins, I want to give a cookie analogy. And so if you think about nice, warm, gooey chocolate chip cookies, those are spike protein. Our recipe is the mRNA. So our mRNA, we basically have the ingredients that we put together to make the batter, to make the cookies. The cookies themselves are the spike protein. We can also think of the oven as the ribosome. So we have all these ingredients we put together, we put it in the ribosome, out comes a nice cookie. The smell of that cookies goes through the house, which is our chemical messengers that we have in our body. So the spike protein is then taken up by antigen presenting cells. And we think of that as like the aroma of the cookies. And so the antigen presenting cells are going throughout the body like, hey, look at this. It's kind of like the smell of the cookies. The whole house is smelling cookies. It's like, man, I really want some cookies. And so we are the T cells and the macrophages. And so we're in the house and we smell these cookies and we're like, I want some cookies. And so we go and we eat the cookies. And then once we eat the cookies, we know what cookies taste like. We know what cookies look like. And if we ever see cookies at the door and we know that they're bad, we can say, we know your cookies, you can't come in here. And the great thing about this is that recipe, the mRNA, is like a Snapchat recipe or like an invisible ink recipe for us older folks that don't even know what Snapchat even is. So once that recipe is read and done, it disappears. That mRNA is out of the body. mRNA does not stay in your body for very long at all. Those cookies, that spike protein, they're gobbled up. They don't stay in your body for very long either. But that immunity that happens, that memory of what cookies smell like and taste like, is in your body for hopefully forever, for at least some determined amount of time. We haven't figured out how long that immunity lasts, but for at least 3 months and hopefully for much longer, we're going to know what those spike proteins look like if they come in our body. And these vaccines are very, very effective. So mRNA vaccine technology has shown to be incredibly effective. When we look at the effectiveness, the red is the infection incidence in people who are vaccinated. And you can see that of vaccinated persons, less than 0.4% were infected. In the placebo group, about 2.4-ish% of patients were infected overall. And this is over a relatively short amount of time. I'm sure we're going to see these data continue to be marched out. And as this pandemic rages on and more cases of COVID are seen, some of those patients were in these clinical trials, and we're going to see even more with more and more patients how effective these vaccines actually are. But in an interim analysis and then with a published analysis of 3 months, it was shown that at the 3-month mark, that as long as patients got at least 2 doses of the vaccine and were not exposed to COVID prior to that point, that this vaccine was about 95% effective for the Pfizer and BioNTech vaccine. And in the Moderna vaccine, it was about 94.5% effective. And so, 94% effective. So these are very, very effective vaccines. These are very promising. I can tell you from personal experience, they produce a very robust immune response. Again, on that day 2, make sure to have your NSAIDs handy, your ibuprofen, your Aleve, because your body, it knows what it is. And so as far as the immune response goes with the virus, we have obviously our innate immunity, which is our barriers to infection. Those don't work very well against SARS-CoV-2. You know, it gets into our nasal cavities, it gets in our lungs, and it causes infection right away. So what we need is humoral immunity and cellular immunity. Humoral immunity is B-cell mediated, and this is where the antibody production happens. And so these vaccines help us to produce antibodies against the spike protein. That those antibodies recognize the spike protein and then present that protein or that virus to our T cells, our macrophages, and other immune recruitment cells, which cause cellular toxicity to the virus itself and protects us from disease. And even in those folks who have received the vaccine, if they get COVID-19, so far, it also seems like the vaccine protects us against severe disease, which is great. And so with that, I'll hand it over to Mr. Jay Singletary. So Jay, do you want me to stop sharing so you can share yours, or do you want me to change the slides for you? How do you want that to go? Jay, I think you're on mute.
You gotta have a mic, don't you? Thanks, I appreciate it, Brian. If you can change the slides, that'd make it easier.
Yeah, sure, I'll just do that for you.
We'll continue. Okay, thank you. I appreciated your presentation. It makes me feel like I want to go home and bake some cookies. So it's been good so far. What I'd like to continue on with now is talk about the approval of these vaccines. A lot of people, before I go any further, I'm Jay Singletary from the School of Pharmacy. And I've been teaching here about 9 years, and I also help certify our pharmacy students for immunization. So I'm involved quite a bit with immunization, along with a couple of other professors. So this is one of the things I enjoy talking about. But with vaccine approvals, you hear a lot about phase 1, phase 2, phase 3. This is actually what they are. Your animal studies start first, where the company takes the vaccine and tries to stimulate antibodies in animals and also look for any adverse side effects. Then they move to phase 1, which is a small number of patients, usually less than 100. The goal here is to determine if there's an antibody response in humans, and also look for major side effect issues. Phase 2 looks at a larger number of patients, still less than 100, but they're really looking at safety and the antibody response. They may also look at tweaking the doses at some point in time. Then you go on to phase 3, which are the big ones, where they get into tens of thousands of volunteers who participate in these studies. They're definitely looking for safety and efficacy. The 2 vaccines we have available right now, there were more than 30,000 patients included in the phase 3 of both of those. So they have a lot of data, and also those studies are ongoing. Even though the vaccine's allowed to come on the market, they're still continuing their phase 3 studies in order to get approval. Okay. Next. Here we look at the vaccines that are available and coming in the near future. The Moderna has the messenger RNA spike protein, and it's here. We're using it. The Pfizer BioNTech is the same type of vaccine. It's also here being used. One of those takes a super cold freezer. Another one just takes freezing to store it. And then we may see some differences as the next ones come out. I heard Monday that Johnson & Johnson did a press release that they're getting ready to submit for an emergency authorization on their vaccine. They have an adenovirus spike protein, and if they submit by the end of January, as they say, they will have their vaccine available maybe mid-February. So we're looking at possibly by mid-February having 3 vaccines available. And they said they've stockpiled more than 1 million already. So that would help our supply. Then you have Oxford AstraZeneca. That is being used in the UK and in Europe. So it's being used in millions of patients already. And then the Novavax, which is a nanoparticle spike protein, that's just starting into their phase 3. Okay. Here we look at where we are in the vaccine approval process. There are 43 vaccines that are testing safety and dosage. 21 vaccines are expanded in expanded safety trials. We have 20 vaccines in large-scale efficacy tests. We have 8 vaccines in early or limited use, and we have 2 vaccines that are approved for full use. And then there was 1 that was abandoned. So there's a lot going on in the pipeline to try to bring us the best products possible to treat this disease. One of the reasons we got our vaccines so quickly was that we could use the emergency use authorization. And before they used that, the commissioner of the FDA, Stephen Hahn, and Dr. Peter Marks from the Center of Biologic Evaluation and Research, both of them made a joint statement that they're committed to expediting the development of COVID-19 vaccines, but not at the expense of sound science and decision-making. We will not jeopardize the public's trust in our science-based independent review of these or any vaccines. There's too much at stake. So this is sort of their commitment to do it by the science, not by any public or political pressure. What we look at here is our emergency use authorization. This is the guy that let us take these vaccines and put them right on the market. About 20 years ago, this was added to the Federal Food, Drug, and Cosmetic Act, where the commissioner can allow unapproved medical products or unapproved uses for approved medical products to be used in emergency if they have to diagnose, treat, or prevent life-threatening diseases. And these are conditions or diseases caused by chemical, biological, radiological, or nuclear threat agents where there's no adequate, approved, or available alternatives. So COVID-19 presented a life-threatening disease, and there's nothing that treats it. You can treat some of the symptoms with some of the steroids and the drugs we have. Some of the antivirals help with symptoms, but nothing treats the actual disease except a vaccine. So this was important for them to take what they had and pull it right out onto the market. They went through the phase 3 studies for so many months. Once they had enough data to feel comfortable that it was safe and effective, as Brian showed you earlier, then they let it go on the market. A lot of people want to know what's in these vaccines, and the companies have been very transparent about their formulations. These are on their websites. These ingredients are posted under cdc.gov, where you can look up these agents and see what's located in the products. It has a messenger RNA, of course. It has lipids, which are the little oily globules that Brian mentioned. These transport the messenger RNA into the cells. There's potassium chloride, monobasic potassium phosphate, sodium chloride, dibasic sodium phosphate dehydrate, and sucrose. Many of you will recognize salt and sugar on the list. Hey, we're baking cookies already. So what you might see in this list is some things are obviously missing. There's no aluminum, there's no mercury, and there are no food allergens. So you don't have to worry about egg allergies or plant allergies, that type of problem. So these are aluminum, mercury, and food allergen free. The side effects that you get when you have an immunization, as Brian mentioned earlier with his, is an immune response. You've triggered something in the body, it responds to it. It may cause some soreness, like the injection site reaction is the #1 side effect. That's going to be a little soreness, maybe redness. There may even be some swelling. There's fatigue, there's headache, there's muscle pain, chills, joint pain, and fever. You may have some or none of these when you get a vaccination, but these are all responses the body has to something that is being generated by the vaccine. These are side effects that are called immune responses. And this is an immune response without getting the infection. That's what's great about vaccines. It gives you this response that causes you to build the antibodies, and it's a good thing. Why should you get a COVID-19 vaccination? Well, you've got these short-term, easily managed side effects. As Brian mentioned, you may need some ibuprofen or acetaminophen, but much less risky than being intubated in the ICU. And if you're taking a chance with a COVID-19 infection, that might happen. We look at survival versus thriving as incredibly different. You can catch the COVID-19 infection, you can try to beat it, work it off, you survive it, and you move forward. But if you're thriving, you step forward, you're positive, you get your vaccine, you do all the things you're supposed to do, and we move forward with eradicating this problem. COVID-19 symptoms can persist for months. They have a syndrome called long haul, LONGHAUL syndrome, where patients get problems with their lungs, their heart, or their brain. It just continues. It doesn't go away. They don't know how long it's going to last and to what extent patients are going to deal with it. So this isn't something sometimes that's one and done. You may have to deal with symptoms of COVID-19 for a long period of time. You have short-term side effects that you deal with. You have 95% vaccine efficacy, and you weigh that against possible long-term health damage from COVID-19 infection. So I think that if you weigh risk benefit, certainly the vaccine is the way to go. The vaccine benefits, as I mentioned, outweigh the known risk of contracting COVID-19. When the vaccine reaches your group, please get the vaccine. They're starting to increase the supply of the vaccine to the public. They're also increasing the groups that are going to receive it. They just gave word that everyone 65 and above can begin to look at receiving their vaccine. One thing people may not realize about vaccines is this is a process. You're going to get your first vaccination. You come back in a month, you get your next vaccination. And then in about 2 weeks, you have your major immunity. So this is a 6-week process. So that's why you want to start it and keep the process going until you get complete immunity. So it takes about 6 weeks. The sooner we vaccinate and establish herd immunity, which is roughly 70% of the population is immune or has vaccine, the sooner that we can remove our mask and hug our friends. As Brian mentioned earlier, someone said the virus comes to us on the breath of our family, the breath of our friends, our associates, and those that are in our circle. So even after you get the vaccine and you have immunity, you can transmit from one person to the next. So you need to wear your mask, keep the social distancing, and also do the hand washing. And this way, I think we will defeat this problem. Thanks very much.
All right. Thank you guys for joining. If it's okay, Sterling, we got a few questions that I felt might be better to give, an answer verbally instead of typing, because it would take a little while. You want me to go ahead and start getting to typing? Into the questions?
Yeah, absolutely. A couple here that were thrown out about the adenovirus.
Yeah, so I typed some
spike
answers.
protein?
So I typed some answers for some of these as we were going through. So the adenovirus is just a shell. I almost think of it like the lipid layer that the Moderna and Pfizer use. They use a viral shell because viruses are pretty good at getting into human cells. And so they use an adenovirus shell to release the antigen into the cell. It comes as, I believe, a little bit of genetic material, but it doesn't have that self-replicating technology. And so I believe each cell that gets infected, quote unquote, with this shell virus makes maybe a few spike proteins, and that's it. So a little bit less of protein production than maybe the standard mRNA vaccines. But it's very standard to use different viral shells that are maybe even more infective than the current virus you're immunizing against, to allow for better entry of that vaccine into our human cells.
Go ahead.
No, I was going to say another question that we got was about allergies, and the CDC has a really good page. Actually, the CDC has a great website in general about these vaccines and answer a lot of questions. And so if we don't get to any today and you still have questions, the website is really, really great. With allergies, so polyethylene glycol, polysorbate 80, and a previous reaction to a COVID vaccine. So if you get one dose of the COVID vaccine and have a reaction, those three scenarios are reasons to not get a second COVID vaccine, or in the case of polyethylene glycol or polysorbate, potentially not even to get the first vaccine for these specific vaccines. I believe there are some vaccines coming that don't have polyethylene glycol or polysorbate, and that would be probably safer to wait for those vaccines. And everyone who receives a vaccine should be monitored for at least 15 minutes afterwards. And if you have a previous allergic reaction to anything, you should be monitored for 30 minutes, just in case.
Any information on the safety of the vaccine for those who are immune compromised?
I haven't really seen any. Jay, have you seen anything?
I haven't seen anything. I know cancer patients are receiving the vaccine. So it seems to be extremely safe with people that have immune compromised situations, but I haven't heard of any contraindications.
Yeah, and none of these vaccines are live vaccines. I think that live vaccine technology, live attenuated vaccine technology would be far too dangerous to do in a very quick time frame. And especially with a virus we don't know all that much about. And so all of these virus vaccines are an activated vaccine, which means that there is no risk at all of the virus causing infection. The vaccine causing infection, which means that it should be safe in immunocompromised populations. What I've not seen, and it may be out there, I just haven't seen it, is any specific recommendations of, for instance, someone who's undergoing chemotherapy, what the time frame is of when you should be immunized relative to your chemotherapy. And that is probably a discussion that should be had with an oncologist. If you have chronic immunocompromising conditions, maybe you're on something like Humira or one of those immunocompromising medications. To my knowledge, that is not any type of contraindication to the vaccine, but always talk to your doctor if you have any concerns.
Had a question here about the vaccine in combating against the new, more transmissible strains of COVID that are starting to pop up in different areas. Yeah,
so it's unclear as of right now exactly how effective our current antibody production is against these new strains with the vaccine immunity. It seems as though the strain out of the UK seems to be we're good to go with the vaccine, that the vaccine should protect against that one. The one that's coming out of South Africa, I don't know that I've heard a conclusive answer on that. And so if that strain does end up coming to the US in large amounts, it could potentially be an escape virus, meaning that the vaccine may not cover it fully, but that has not, to my knowledge, been answered definitively. The UK strain seems to be good to go with the vaccine, and that, from my knowledge, seems to be the one that we're seeing in the US, and we haven't seen the South Africa strain quite as much. But it's still really, really new, and there hasn't been a lot of exposure to vaccinated patients with those strains. And so I think it's really too early to make conclusions either way. Jay, I don't know if you've heard anything else besides that.
Just, I know Dr. Fauci said that the small changes on the spike don't seem to be enough to affect the vaccines that he's seen. But again, they're watching the data, but he felt comfortable we were okay.
Mm-hmm.
You mentioned someone who has had the vaccine will test positive after receiving it. How should this be managed? And does this mean someone who has had the vaccine cannot be tested, but also could be spreading it at the same time?
So no, the testing that could be positive would be an antibody test, which is a blood test. The antigen test that's used, there has been no test on the market currently that has shown any cross-reactivity with the vaccine. So even if you just got the vaccine, yesterday or the day before, if you got it a month ago, there should be no cross-reactivity. If you had the vaccine and you have an antigen test positive for COVID, so a nasal swab test for COVID, you have COVID. So that's not the vaccine. Don't blow that off. 5% of patients in these vaccine trials did get COVID-19. They tended to be more mild cases, which is another benefit of vaccination, that seems to spare folks, at least with the data we have, seems to spare folks from severe disease from COVID-19. But if you test positive on a nasal swab or one of those deeper tests, if that comes up positive for COVID, you have COVID. I mean, I guess it could be a false positive, but it's not because of the vaccine.
And Brian, I think that's an important point, and we want to thank Elizabeth Marnick because the faculty had requested last summer that I always include messages that went out to students and attach them to the faculty updates, and so I did that this week. And so Elizabeth brought to our attention that we had some misrepresenting information into that letter that went to students. And so I have been in contact with Troy Morehouse. I think he's connected today on this session. And I asked Troy to double check with the Broad Institute, which is where we do our student testing. And as Elizabeth suggested, they've indicated that for all of our Broad testing, that wouldn't be impacted. So as Nancy was suggesting, does that mean that students wouldn't be able to go into our testing cycle? But we do have have some students who have part-time jobs or in locations where they're not allowing for rapid tests. And so we will be sending out another letter through Troy's and Pam's office this week on, I think, Thursday. Eric Gordon's working on that with them, that will clarify that differential. So we apologize for that misrepresentation, and certainly appreciate Elizabeth quickly bringing it to our attention. But we do plan to have students in that testing cycle. One of the things that we did want to do and did include in that letter was because students have been remote for the last 2 months, there may be students who tested positive for COVID. And so we want them disclosing that to us, so we're not getting elevated pieces, because they could have antibodies for up to 90 days. And so the same thing for employees. We have a pretty good count with our employees because they've been connected, but we may have some students who just haven't informed us that they got COVID over break. And so we will want to make sure that we're trying to ask those specific questions as part of our wellness testing cycle. We are going to continue to have that ramped up at that same high level that we did in Thanksgiving, right before Thanksgiving break. We increased that in November, and we'll stay at that increased cycle level so that we're trying to get students through that cycle every couple weeks. And then we will be following the same practices that the University of Maine system and most of the privates are, that is also included in that letter about asking students who are coming from outside of the state to make sure that they're showing testing before they come back onto campus.
Perfect.
Question here, this is an interesting one. If you got a vaccine, you got the first vaccine from one manufacturer, and the second dose is not available from the same manufacturer, is it effective to receive the second dose from another manufacturer? So if you had Pfizer first and then it wasn't available and you had to go to Moderna, is that an option, or should you avoid
that? You want to take this one, Jay?
I mean, I really haven't heard anything on that. If there's nothing else available, I certainly would use the other brand. But
I believe the current recommendation as of right now is that if you're vaccinated with one specific brand, to stay with that same one.
right.
They may change that guidance in the future. But as of right now, the recommendation, if you start with Moderna, do the Moderna. If you do Pfizer, do Pfizer. They are different doses of antigen. They are different in some of their ways. I mean, would the immune response be the same between the two? Almost certainly. But as of right now, that is not recommended to be done to cross vaccines at this point. They may change that in the future.
Trying to kind of avoid some that are somewhat repetitive here, so I'm going to try to kind of bundle a few of these into one question. What are you hearing about the different waves of giving out the vaccines, how that's going to work, kind of what is included in wave number 1A, 1B, and what does that timeline sort of look like?
So each state's going to be different, and there are websites that go into the rollout for each of these. I don't have it bookmarked on mine because selfishly I already got my vaccine. If I hadn't gotten it, I would be finding this website and be looking at it every day. And so I don't know if someone has the link, but if you do, please post it in the chat box. But I can certainly try to find it. And for Maine, we're a little behind other states because we weren't quite as big of an epicenter. And so some states are already vaccinating elderly, for instance. But I think I saw in the chat box, I believe it was Wen that said that we're going to be doing that for Maine, or more information coming on January 18th. So we might be entering phase 1B pretty soon. Which I hope is the case because, when it comes down to it, the frontline workers, I am very, very close to COVID patients every single weekday. And so it is good for me to get the vaccine so I don't get it. But if I did, honestly, I'm not a huge risk for having severe disease. These patients that are 70 plus are very, very high risk. Our COVID units in the ICU are mostly patients 70 and above. Well, it's actually not all the way true, but maybe 60 and above. And so, those patients from a disease severity standpoint really need this vaccine, probably more so than a lot of us on the front lines. So I hope that we can start vaccinating them much quicker.
Due to how new this vaccine is, any information about how long the immunity lasts with an mRNA vaccine would it be better off waiting to take one of the vaccines that use the COVID-19 virus as part of creating the vaccine in order to develop long-term immunity?
There's no indication right now that other vaccines are better. In fact, preliminary data from the AstraZeneca vaccine actually, or was it? Yeah, I think it was AstraZeneca Oxford vaccine, actually shows that it may be less effective than the mRNA vaccines. Again, this is very early data. So as the studies go on, those numbers are likely to change. But they're certainly not more effective than the mRNA vaccine. We don't have enough data for long-term projection of anything, of how long immunity lasts with these vaccines. So for any of the vaccines, we have absolutely no idea how long immunity is going to last. Even for natural infection, we still don't have a great handle on how long it will last. There are some patients that have gotten a reinfection within as short of a time frame as 3 months when they were infected with COVID, and then they got COVID again 3 months later. Others, have not had a second infection. Many others have not. And so for some individuals, they may have much longer lasting immunity than others. This could be a seasonal vaccine like the influenza vaccine. I've heard that tossed around, but at this point, it's far, far, far too early to really have any long-term idea of any of this. Based on the history of vaccines, though, long-term safety data is probably going to be safe because almost all vaccine reactions, at least that we know of, happen within about 3 months of the vaccination. And all vaccines, before they were allowed to enter an EUA, had to have 3 months of safety data before they were approved. And so I don't have any long-term safety concerns with these, even though it's a new technology, because like I said, mRNA does not stick around the cells. It's not going to be there mutating your DNA or anything like that. You may have heard that, maybe some of these alternative social media sites. But I can say with all certainty that this vaccine doesn't hang around your body for very long. And the incidence of adverse effects outside of, influenza-like symptoms for a day or so seems to be similar to baseline. There's a possible signal of a slight increase in Bell's palsy, which is like an eye drooping, facial drooping kind of thing that is almost always reversible, can happen with natural infection. So it's unclear as to how vaccinated patients versus infected naturally patients will have as far as the incidence of Bell's palsy. But I know me personally, I'll take 100 cases of Bell's palsy before I would take a COVID infection. And the overall rate of that adverse effect is extremely low and barely above the population baseline, even without viral infections.
Have you heard of any negative interactions with other vaccines? Example, shingles, flu, etc.
No. When I actually did an annual checkup at my doctor the other day, and just was curious as to what they're telling their patients, and they're saying to space out vaccines by about 2 weeks. I don't know that that's really based on anything. Since it's an inactivated vaccine, it shouldn't really matter. It shouldn't interact with anything else. But because it can lead to some pretty intense like flu-like symptoms, potentially, it might be worth not doing any other vaccines on the same day you're getting your COVID vaccine.
I got this question here. If I hit the pronunciation, I'm going to sound really smart. So here goes. What is known about the efficacy of the vaccine when taken concurrently with immunosuppressant medications? Is there any way of knowing the efficacy after the 2 injections?
Those patients weren't really included in the clinical trials. So I can't say for sure. Certain immunosuppressing medications may have a bigger effect than others. Chemotherapy comes to mind, and that would certainly be one to think about delaying until you have recovery of your bone marrow. I'm not sure about that. Some of the other things that have come up are like Tylenol or acetaminophen and NSAIDs like ibuprofen. It's not recommended to take a dose of those before your vaccine. So don't take like a Tylenol or an ibuprofen like right before you go in for your vaccine appointment, because that could potentially, based on old data, might impair the vaccine effectiveness. But it seems to be very safe to take either acetaminophen or NSAIDs after the vaccine has been administered. And I know the general consensus among like the med Twitter and stuff like that is that symptoms tend to occur about 12 hours or so after that second vaccination. Those tend to be the time where most people get those side effects. Mine happened, about 12 hours after, and then ramped up, and about 24 hours after, as I finally broke down and took some ibuprofen because I had a headache. And after I took that, all the symptoms went away. They haven't come back. So,
What is the biggest difference between the first and second shot of the vaccination?
So the first shot, your body hasn't seen the spike protein before. And so you mount a little bit of an immune response, probably a good enough immune response to have some efficacy of preventing infection. But the second dose, your body's already seen it. So think about like if cookies, in the cookies analogy, your people in your house are like adamant fitness, like, no sugar in my body at all. They're going to take that person that comes to your house with a plate of cookies. They're going to slap those cookies out of their hands because it's like, I know what those are. They're cookies and I don't want them. And so that immune response is going to be so much more on that second dose because your body knows what that spike protein is. And so when it sees it for the second time, all your inflammation goes in there. So it's actually a good thing, as Jay was saying, that's a good thing that your body reacts that much because it shows that your immune system is functioning well. Now, if you don't get those symptoms, it doesn't mean that your immune system isn't working because only about 50% or so of people had those types of symptoms in the clinical trials, and the efficacy was 95%. So obviously there's a disconnect there. But if it happens, it means you got a working immune system. Jay, you want to add anything to that?
No, I think that's exactly right on.
We got two more and we'll wrap up here. What is the data on the number of vaccinations offered versus those accepted or taken so far?
I don't know. Have you seen any of those statistics, Jay?
I have not, no.
I've heard that in the healthcare workers specifically, and these are not usually like folks that are taking care of COVID patients. These are usually more ancillary staff, things of that nature, that the vaccine acceptance rate in some areas is as low as like 60%, of people who are eligible that actually get it. And so there are a lot of people nervous about this. We didn't go into the approval process because it is a much shorter time frame than usual. But one of the things that because I'm a big skeptic. So this mRNA vaccine, I was like, we've never done this before. So like we might be growing like third legs and stuff. Like I don't want to just take this. But when I looked at the approval process, what they actually have done is cut out most of the bureaucratic steps. The reason that vaccine approvals take so long in general is there's a couple of reasons. One is that there's a lot of red tape, and a lot of that red tape was cut down. And so like what normally would have taken like a 2-year review was done by a lot more people in a short amount of time. So it was divvied up, made more efficient, et cetera. Another reason why vaccine approvals sometimes take a long time is because you don't have enough incidence of infection. So if you think about something like Haemophilus influenza, which we developed a vaccine for that in the mid-90s, Haemophilus influenza B does not occur all that often, even without vaccination. There were about 20,000 cases a year in the US. So you are just counting on the fact that the patients who got vaccinated and those that got placebo are going to be exposed to Haemophilus influenza and either get the infection or not. Because you have to have a certain incidence rate, a certain number of infections before you can claim efficacy. In the case of COVID, because we're in a pandemic, we have a lot of infections. And so that incidence rate happened very quickly. I mean, you saw within 3 months, there was a huge difference in the number infected and the number not in the vaccinated versus placebo arm. And that's because there were a lot of circulating COVID cases that allowed the vaccine efficacy to be shown in a much shorter time frame than usual because we're not usually developing vaccines in a pandemic. And so because of all those reasons, it was able to be done. And also the government funded the production of these vaccines in the background so that if these vaccines ended up being ineffective or unsafe or whatever else, the drug companies wouldn't be on the hook for, a few $100 million of vaccine production. They could, basically the government would pay them for that vaccine. And so the production was going on as the trials were going on, which is another thing that doesn't usually happen. Usually production happens after all the clinical trials are over, after FDA approval, and that's why the vaccine usually takes so long. This is a coordinated effort. It was actually done, in my opinion, in a very, very safe way. And it gives me hope that we can maybe help make vaccines a little faster in the future. It also helps that these mRNA vaccines are very easy to make. One of the questions was if you have an egg allergy, because influenza vaccines are made in eggs. Well, it takes 8 months generally to grow those vaccines in eggs, to at least to scale it up. With mRNA, you can slap those things together pretty quickly. You're just baking a bunch of cookies. And so, yeah, I think this mRNA technology, it looks like it produces a very clear immune response, very easy to make. I can easily see us using mRNA for other vaccines in the future.
Okay, we're approaching the end of the hour. One last question here. You mentioned the difference between disease and infection. Does the vaccine prevent both disease and infection?
So this is a tricky one because it does prevent disease. In at least most of the vaccine trials, they did not come in for routine antigen testing for like screenings, for instance. So it may be possible to be a carrier for the SARS-CoV-2 virus, and you won't develop the disease, but you might be able to transmit it to other folks. We don't have enough information yet as to whether or not these vaccines reduce colonization of the SARS-CoV-2 virus. So everywhere I'm going, even if mask mandates are over, until this COVID-19 pandemic is pretty like over, I'm still going to mask. I'm still going to be very cautious and do distancing around folks, because even though I'm vaccinated, I might still carry the virus in my nose. And if I give someone, COVID-19, I'm going to feel terrible. When I come home from work every day, I immediately change all my clothes. I, wash all my face and hands and arms and everything that could have been exposed to virus, because I don't know for sure that I have the ability to stop that colonization. So it certainly is something to think about, and hopefully more data comes with that. But as of right now, there's no indication for or against us reducing colonization of the SARS-CoV-2 virus with vaccination.
Very good. That brings us to the end of the hour.
Brian and Jay, we really appreciate you sharing your expertise with us, and actually to all of the clinical faculty in the School of Pharmacy who have been on the front lines, and especially to Eric Gordon, who spent a lot of time highlighting their work and gotten us some excellent brand elevation at the national press level, with publications, several publications now that have hundreds of thousands of viewers related to the work that our colleagues in clinical pharmacy are doing. And then also want to call attention to some new opportunities that are being provided for our students in nursing for paid clinical internships as part of that vaccine effort too. So it's exciting to see that Husson can take a lead, especially in our community and in our state, but certainly that's also gleaning some national recognition. So we appreciate your time and your willingness to answer so many questions and to share your expertise.